Good afternoon. My name is Tanja and I work for the terrestrial ecology unit of the Department of Biology. And today I'm going to present some of the results of the research I did about the germination and establishment of grassland seeds following endozoochory. So I'll be talking about seed dispersal, and seed dispersal is a very important process in plant ecology as it enables the colonization of new habitats, but it also maintains genetic diversity, which is very much needed for maintaining healthy populations. It is also a main driver of vegetation dynamics and succession. And there are many examples of seed dispersal mechanisms, and most of them need either abiotic or biotic vector. Examples of abiotic vectors might be wind or water, while also animals or even man can disperse large quantities of seeds. And a very special example of animal-mediated dispersal is endozoochory, which implies that seeds are ingested by animals and they are later on defecated as intact and viable seeds. This is a rather complex dispersal mechanism, so there should be many conditions fulfilled in order to make this happen. The first step is that diaspores actively get ingested, but they also need to survive the digestive system, and they need to be deposited on a suitable site where they are able to germinate. So when we look a little bit more into detail, we see that the seed dispersal cycle is parallel with the life cycle of plants. So we start with the availability of fruits, which gets ingested by animals. And whether they get ingested or not depends on the habitat choices of animals, which diets they prefer, also the availability of plants in specific habitats. So once seeds are taken up, they travel through the digestive system of animals, and finally they get deposited in a dung pad on the field. So when and where this happens also is a function of seed passage time, which is very specific for each animal species, but also it depends also on which foodstuff has been eaten. Also, dispersal movements will tell us where we can find seeds at the end of this dispersal process. In some cases, there is a phase 2 dispersal. For example, when dung beetles are active at the site, they can take away little pieces of dung, including those seeds, and put them away somewhere else. But today I will focus on the process of seeds dispersal itself. Once this process is completed, so we have viable seeds, those seeds can either germinate or die. So it's in the best interest of plants, of course, that they germinate, and they grow into seedlings. If they survive, they grow into juvenile plants and eventually adult plants. So the circle is complete and they can start flowering and fruiting again to provide next generations. So I was mainly interested in temperate grassland species because endozoochory is here a widespread phenomenon, as we find many plant species germinating in dung. And this is also a very hidden process because those seeds don't seem to have very apparent adaptations to this dispersal mechanism. They don't have fleshy fruits, for example, to attract animals. And in the foliage as a fruit theory, it is postulated that not the fruits itself attract the animals, but it's rather the plants that are very tasty and highly nutritive that attract animals, and they just accidentally also ingest seeds. So what about temperate grazers? Most of our natural temperate grazers are extinct. So in this research, I used cattle and horses as model organisms because also these animals are commonly introduced in nature management. Sorry. And so it's very important to know what their dispersal potentials are. So I will talk in terms of costs and benefits to quantify this process. I will give the results of 3 experiments I did about the process of seed transfer in the animal, the germination in dung, and the later growth and flowering. So in the first experiment, I tried to simulate each step of the digestive system of cattle and horses. So I did mechanical treatments in which I used real horse and cattle skulls to chew the seeds. But I also used chemical treatments to simulate the digestive fluids which are present in the stomach. I also used temperature treatments and combination of all other processes, and I compared them with a control treatment. I used several typical grassland species in this research, and I measured the germination daily to know whether things get speeded up or not. So, and the main result of this research was that we found an increased germination of hard-seeded species, which are typically very slow germinating. So the idea behind this is that by chewing, that the seed coats get damaged, which increases the germination potential. Also, we found that the chemical treatments increased the germination of grass species, and they are also very commonly found in dung, and this might explain this. And we found rather negative effects of the thermal and combination treatments on the germination of most species. But still, most species had a different response to the treatments. So we found, for example, species which experienced the high costs. For example, clover species germinated slower and less, while stinging nettles, for example, are very successful endozoochory species because they germinated even better after those treatments. So what goes next once seeds are deposited in dung? So I did a second experiment, in which I tested the effects of cattle and horse dung on the germination of the same grassland species. And here the results were far more straightforward. Dung decelerates and reduces germination of all species. And even in cattle dung, we found more pronounced effects, which can be explained by the fact that cattle dung has the typical trait that it develops a hard and dry crust very soon after deposition, and this crust is very likely inhibiting the establishment of the seedlings. Also, the phytotoxic compounds that are commonly found in dung might have an inhibitory effect on the early life stages of seedlings. So we found very high costs. But also we need to take into account whether this is beneficial or not in the later life cycles of plants. It is not always a very a good trait to germinate early because you have a high competition, the conditions might not be good, and also there might be effects of dung in later life stages. So I did an experiment on the growth and flowering in dung using the same species and the same dung species. And here we didn't find very conclusive differences between horse and cattle dung, but we did find a clear effect of dung on the growth and flowering of the tested species. In the seedling phase, we didn't have too much effect, but in the growth and flowering phase, we had clear differences, as we found higher and heavier plants after application of dung, and also they had more flowers. So here we had clear benefits. So to summarize, I present you the list of species that I tested in the several experiments that I conducted. And we see a clear species-dependent response. So we see a high cost in the germination phase, especially in the establishment phase in dung. In the seedling phase, we see few effects, while we find major benefits in later life stages of growth and flowering. So I thank you all for your attention.
Questions?